So our first session today is Diagnostic Advances Beyond the Genome. So we've got some great speakers. We've got Dr. Jamie Ellingford, who's going to talk about transcriptomics. We've got Dr. Julia Carrasco-Zanni-Sanchez talking about proteomics. We've got Professor Bill Griffiths here to talk about metabolomics, and Dr. Nikolas Pontikos to talk about artificial intelligence. But when we think about diagnostics, many of you will have heard about the advances in diagnostics with respect to genomics, so making genetic diagnosis for rare conditions. And this area of work has really advanced over the last decade. And we'll be covering some of that in our breakout session later. We've covered some of that at Rare Summit in the past. So what we wanted to touch on first here today was thinking beyond the genome and thinking about some of the other technologies that can also be informative in helping to make diagnoses for individuals with rare conditions. And the reason for this is because even with whole genome sequencing, we're making a diagnosis for only around 30% of people with a rare condition. So we've come a long way, but there's a long way to go. So why is that? Well, some rare conditions are not genetic, so genomics therefore won't help in making a diagnosis. But most of them are. But even with that case, we're still missing many of these. And there are lots of reasons why that's the case, and they kind of fall into two camps. So on the one hand, we have issues with technology. So some areas of the genome are difficult to sequence accurately. And we have to interpret our genetic information using bioinformatic pipelines. And these pipelines are being improved all the time, but they're not perfect. So there are issues with technology. But there are also issues with our knowledge. So we don't yet know what all genes do. And we really can't interpret all the different genetic changes that we find in genomes. So there's lots of opportunities to improve sequencing technologies, to improve our bioinformatic pipelines. And there's lots of opportunities with respect to genetic and genomic data. We can get more data. It will help us interpret the data. If we share our data, that will also help. And these types of approaches will be covered in our Better Genomics breakout session later. So, however, there are also some complementary information that can be gathered from other technologies that can help us to make diagnoses for genetic conditions. And that's what this session is all around today. So, transcriptomics, proteomics, metabolomics, and AI. You may be really, really familiar with these terms. Think about them all the time, think about how you can use them. Or you may be feeling a bit more like Dorothy and Friends, like lions and tigers and bears, oh my, these are really long words, and what do they mean? So I just thought it would probably be worth giving you a little bit of context of these terms before we start and introduce the speakers, just to explain what they mean. So we're going to walk from our genetic code through to what we would describe as a phenotype, which is how that condition manifests in the person. So we'll start with our genetic code. Our genetic code, or our genes, are encoded in a chemical called DNA. And so that's our instruction manual for our bodies, for helping them grow and function. When we study that instruction manual, when we study our DNA, we call that genomics. So when we sequence DNA and when we try and analyze it, that is genomics. So that's your first omics. Omics just means you're doing a lot of it. It means you're doing it at scale. Okay. The DNA is an instruction manual. It doesn't do anything. It is instructions. So the first stage in biology from that instruction manual is to carry that instruction out of the nucleus of the cell so that it can be read by the cell. And that is done with a different chemical called RNA, or ribonucleic acid. When we study RNA, that is called transcriptomics. Okay, so that's the first thing we're going to think about today. Then we use that instruction to make something. So in crudest terms, our bodies make proteins from the RNA instructions. and proteins are the things in our bodies that do things. So when we study proteins at scale, when we analyze their structure and what they do, that field is called proteomics. The proteins do things, and they do things that produce other chemicals in our body, and we refer to those as metabolites. So when we study metabolites, we are doing metabolomics. And eventually we get to what this means for a person, what their phenotype is or what their clinical features are. So what is seen in a patient or what is seen in a person who's affected by a rare condition. So that's their phenotype. And that can be, what we're going to do today is we're going to talk about how artificial intelligence can help inform about phenotype. Now, artificial intelligence can be used for all of these steps of the process, but we're going to focus today on its uses in phenotypic terms. So with that, I'd like to just welcome up our first speaker, Dr. Jamie Ellingford, who's going to talk about transcriptomics.
Thank you very much, Gemma, and thank you everyone for being here and for the opportunity to speak to you. So I'm Jamie Ellingford. I am very fortunate in that I get to wear kind of two hats in my... Oh, I'm showing... There we go. I get to wear two hats. So I work for Genomics England, where I play the role of lead genomic data scientist, and so help develop some of those bioinformatic pipelines that we apply to genome data to help diagnose rare disease. And in the other part of my life, I'm also a research fellow at the University of Manchester and lead a research team that's very much focused on how we use transcriptomics, or the sequencing of RNA, to understand about cell biology, but also to understand about how genomic variants give rise to particular genetic conditions. So the things that I'm going to talk about today aren't attached to either of those things. It's really my viewpoints of kind of where transcriptomics sits. I'm going to talk a little bit about kind of some of the promises, give you some examples, but also speculate about some of the challenges about integrating these technologies in a routine diagnostic setting. So as Gemma's already introduced, we have this central dogma of molecular biology, where this was the original figure produced by Francis Crick and from his work in the 1960s, where we have this transition of molecules from DNA to RNA to protein. And where we're going to focus in this session is very much at that RNA level. And so transcriptomics is the surveillance of all, and I've put all in brackets there because as much as we can see for the particular cell type that we're looking at a particular time that we're looking at it, but all the RNA that's possible within that particular context. And so we're going to come back to that in a moment, but essentially we're trying to look at which genes are being turned on at that particular time point. So if you've ever seen kind of the content of a genome before, then it's quite staggering. So it's 3.5 or 3.4 billion letters long, so individual nucleotides. And if we were to look at any one individual in this room, we'd have something like 5 million different genetic variants to the reference genome that we use as kind of the basis of what a human genome should look like. And I use those terms very, very carefully. But one of the staggering things about it is that only about 2% of those 3 billion nucleotides or so actually directly code for protein and go through all the way through that transition from DNA to RNA to protein. The other 98% was once termed junk DNA, but we know that it has really important functions in understanding how genes are turned on, when they're turned on, and precisely which parts of those genes make their way to become proteins. And so a slightly different way of presenting this is in this cartoon here, where the thick green boxes, they represent the coding part of this particular gene. But then there's lots of other things that we've annotated there, and that includes enhancers, promoters, intronic regions, untranslated regions which make their way to the mature molecule. And they're all kind of complex concepts that we can't go over in detail right now. But all of these particular regions are important in controlling where a gene is expressed, how much of it is being expressed, and precisely what content actually makes it to that protein. And the more and more we look, so with the opportunity to mine whole genome data, both in diagnostics and in a research setting, we see that genomic variation can disrupt that normal process and can lead and drive some particular genomic disorders. And so one of the important things that we can do with transcriptomics is go kind of the next step. So from a genome, we can see that these variations exist. We can potentially predict the effect that they'll have. But in a diagnostic setting, at the moment, in 2023, that certainly isn't good enough to provide a confirmed diagnosis. But transcriptomics, the study of RNA, can kind of prove the effects of these variants. And so as a result, give rise to diagnoses, and that can, as I'm sure you're all aware, be transformational for families that receive that diagnosis. And so I wanted to give you a couple of examples. So on the top here, you see an example of a deeply intronic, so a variant that sits between the coding parts of a gene in the BRCA1 gene. So this is a predisposition to cancer gene. And the data that I'm showing you, so at the top here, the red bits, that's the RNA that we've sequenced from a patient. And at the bottom here, you can see the blue bits are the RNA that we sequenced from a control. And the important thing about these diagrams is the lines that link things together. And so in the patient example, you can see that there's lines at the top here, and these big peaks, these are the exons, these are the protein coding part of the gene that eventually becomes protein. But you can also see this line at the bottom here that's linking together and skipping out part of that protein coding sequence. When we look at the control sample, you see that we don't have this line which links this kind of site here to this site at the bottom. And so what we're seeing here is an exon skip. And so part of the protein coding region that should be present for BRCA1 in this case isn't making its way all the way through to the protein. And as a result, this can often cause in kind of the loss of the function of that particular allele. In a different example, down here, you can kind of see the reverse. So we see two exons at either side of this diagram. And in the control sample, you can see that these are linked. And so that's all that's being expressed. Whereas in the patient sample here, you can see that there's part of this intron, which is also being expressed in the RNA. And so this is a pseudo-exon inclusion. Part of the intronic sequence of this gene is now being included as part of that message that then gets converted into protein. And again, this has a very similar effect in that it can often cause the complete loss of function of that allele. So that's two examples of many that we could pick out where sequencing of the RNA, being able to develop a transcriptome, can be transformative for a diagnosis because we prove the functional impact of these genomic variants that we've identified. And so I'm going to skip over that example and just kind of think about some of the challenges that we can go into in more detail in the discussion. So I wanted to pick out some of the major challenges from my point of view. So the first is that we've already seen that the non-coding genome is a massive search space, and most of the genomic variation that we have ourselves exists in that space. But until recently, we haven't had any clear guidelines about how to analyse these things clinically. Very, very recently, we've released a draft set of guidelines to try and help with that. They're very much draft, and we want to see how these work in real-life context, to understand the limitations, to understand kind of where there's benefit to applying these guidelines in routine diagnostics. But that's only part of the puzzle. That highlights variance. It doesn't necessarily give that functional impact and enable genetic diagnoses. The second thing we've already alluded to, some of these regions that are important for when a gene is switched on and where it's switched on differ between cell types and differ over time. And so the actual RNA or the regions of the genome that we think are important to analyse may differ by different cell types, and that itself presents lots of challenges. The third, as we've just stated, RNA is switched on at different times in different places, and so actually the samples that we take from individuals mean that in some cases it's not appropriate data that we can generate to survey particular genes. And so one of the PhD students who was with me a couple of years ago developed a tool to predict which genes are possible to survey from particular tissues. And so we looked at blood, at lymphocyte cell lines, to cell lines that we can create in the lab, as well as a couple of other clinically accessible things. And the reality is it's really quite difficult. So if you've got a gene which causes a retinal dystrophy, it's only expressed in the retina in the back of your eye, then the likelihood is that that's probably only switched on in that particular tissue. And so actually accessing tissue that's representative of that is really quite difficult. And finally, we have no kind of standardized approaches for, firstly, how we generate that data. Lots of different technologies exist, the main distinction really is whether we do short read or long read sequencing, but also the bioinformatics and the data analysis approaches that we apply are also can give different answers, and they're not really standardized in a centralized way. And so as part of my research team, I'm working closely with teams in the NHS who are starting to develop this technology and come out with standardized approach for both extracting RNA as well as sequencing it and data analysis. And so that's very much in its early stages, but it's something that we hope will make its way to the clinic in the future. So I'm going to stop there, and we can elaborate on any of those ideas in the discussion session, but thank you very much.
Okay, I'm going to ask Julia to come up now and move on to the next stage of the process, which is proteomics.
Thank you. Okay, great. Hi, everyone. And again, thank you for the opportunity to present some ideas here today. So, we're going to be talking about proteins in this part of the session. And, both Gemma and Jamie have already walked you through this bit. So right now we're kind of focusing on this step. And the crucial bit to remember is that proteins are, the functional readout of both the genome and the transcriptome. So they're actually, the molecules responsible for doing things and carrying all the functions that your cells and your bodies need. And they're actually really attractive for diagnoses and in clinical practice, because even though they're produced in different organs around your bodies, a lot of them actually end up circulating in your blood. And what this means is that it's really practical and convenient to be able to, have a patient come into the doctor, take a blood sample, and then trying to measure different proteins. Having said that, the field of proteomics, and particularly for diagnoses of rare diseases, is in its relatively early stages. And one of the main reasons for this is because, unlike some of the other technologies that we're discussing in the panel, measuring many different proteins in a blood sample is actually technically very challenging. And therefore, it also has high costs associated with this. So what this means is that even only a few years ago, we were in this scenario here, where we could only measure very few proteins found in very high abundance in your blood, and we could only do this in few samples. But thanks to some recent technological advancements, we are now in this scenario where we can actually measure thousands of different proteins that are found circulating in your blood, and we can do this in many, many more samples. So we can really scale up and then do bigger studies to try to understand how this can help in diagnoses of rare diseases. So I'm going to walk you through a couple of examples of some of the things that people are working on in the field and how this can potentially help. So the first one is perhaps one that most people in the audience will be familiar with. So when we're using proteins to try to inform on certain genetic diagnoses. So let's say we have a patient with a rare condition, and we've done whole exome or whole genome sequencing. And we have identified a mutation in a gene which is considered to be of relevance for the specific disease, but we're actually not entirely sure whether the specific mutation is being damaging to the protein function and therefore potentially the cause of the disease. So one thing we can do in this scenario is to take a blood sample, measure the proteins, and then go specifically look at the levels of the protein that is produced by the gene carrying the mutation, and then compare this against the levels of the same protein that we see in people without the disease. And if we see that the levels in our patient are drastically different, then we can hypothesize that probably this mutation is having a damaging effect on the protein. Now, the second potential application is on actually using proteins themselves as diagnostic markers. The idea here is to basically try to find proteins which we can only detect in blood in response to specific disease processes. So the idea here is that when we have a patient and they start presenting with symptoms, we could take a blood sample, and if we detect this protein, then we can say with some degree of confidence that this is a way to make a diagnosis. But sometimes actually one protein does not give us sufficient confidence to be able to make these diagnoses. And one of the strategies that are being followed in the field is to try to find several of these proteins and try to build what we call a protein signature. And then this tries to give a little bit more confidence in trying to make these types of diagnoses. And one thing that is really attractive about this is that unlike your genetics, which is mostly fixed throughout your lifetime, proteins are very dynamic, and they actually change throughout your lifetime in response to both genetic and environmental influences. So one of the other potential applications leveraging this is that some of these diagnostic proteins can actually also serve as surveillance markers. So for example, if we have a patient with a history or a family history of a specific rare disorder, some of these proteins can be used to try to take blood samples even before the onset of overt symptoms, and to try to use the levels that we detect to make sort of an educated guess or a prediction of how likely it is for this person to start developing symptoms within the next, let's say, 5 to 10 years. However, one caveat of this approach is that actually finding these diagnostic markers is tremendously difficult. We need very complex study designs in which we need to recruit many people who are affected by the same condition and many healthy volunteers, and it's a quite complex endeavor to try to find these markers. And then finally, just to finish up, the third example is in trying to use proteins to refine diagnoses. So perhaps some people in the audience may be familiar with, even though two patients may have exactly the same diagnoses, they actually experience a heterogeneous range of symptoms. And sometimes there is an underlying reason for this, and this may be that some of the mechanisms or the biological pathways that are affected are different. So the idea here is that if we can try to find some proteins that are sort of tagging these different altered mechanisms, we can perhaps try to allocate people into specific subgroups. And the question is, why is this helpful? Well, because then when we try to make new diagnoses and we try to allocate people into these different subgroups, we can perhaps be a little bit more or have a better idea of the types of symptoms that the patients are more likely to experience. And this can inform the sort of follow-up and clinical care that they may need. So just to briefly kind of summarize, I've shown you some of the opportunities in the field and some of the examples that people are working on. But as I said, it's relatively early days in this field, mostly because of several technological challenges associated with this technology. So we're only kind of starting to understand the diagnostic yields that could be achieved with this technology. But having said that, these technological advancements are happening at a really fast pace, and it's quite an exciting time for proteomics and the possibilities in diagnosis of rare diseases. Thank you.
I will try and introduce you to metabolomics and lipidomics and how they may be relevant in the context of rare diseases. And before I should start, I should thank the organisers for the invitation to come to speak to you. Okay, so as Gemma said, really, metabolites are the small molecules that actually we make in our bodies. So they might be signalling molecules like hormones, they might be structural molecules, they might be breakdown products. But the key is that we actually make them in the body. So my favourite small molecule is this one. This is cholesterol. So it's a metabolite, but also it's a lipid. So lipids are fats. So lipids are actually a subsection of the metabolites. So a key point is that the pattern of metabolites and lipids that we see in our body fluids very, very often change in response to a disease. So we might have, if we have a particular disease, that disease might be reflected in our metabolic pattern in our body fluids. So this leads us to the subjects of metabolomics and lipidomics. So metabolomics really, it's about measuring the metabolites in a body fluid. And then lipidomics, as I said, that's a subsection of metabolomics, and it's specifically measuring the lipid contents of a biofluid. And we do this in the hope that we can actually help in making a diagnosis of a disease, maybe a rare disease. So let's just walk you through how these metabolomics and lipidomics work. So we start on the left here with our sample, which is usually plasma or urine, or it could be cerebrospinal fluid. We then separate out the different molecules in the second panel on what we call a liquid chromatography instrument. So that just separates out the molecules. They then go into another instrument, which is called a mass spectrometer. And the mass spectrometer allows us to identify the different molecules, determine their chemical structures, and also measure their concentrations in our sample. And a mass spectrum looks like this in the central the third panel. All those peaks you see, that's a typical mass spectrum. And each one of those molecules, each one of those peaks will be a different molecule. And the height of the peaks is going to be proportional to the concentration of the molecules in our sample. So we can identify the molecules in our sample, and we can determine the concentrations. Once we've identified the molecules, we then try and put them onto what we call a metabolic pathway. So a metabolic pathway is really a reaction scheme of how different molecules are made in our body and then broken down. So on the right, we've got a metabolic pathway. Now, if we've got a rare disease which results in an error in one of the reactions in this metabolic pathway, so maybe one of the reactions in this metabolic pathway is not working correctly. So here I've got my red line. So this is where a reaction is not working correctly. When we have that, then we then see a build-up in the metabolites above the red line and a fall in the metabolites below the red line. Now, metabolomics can then measure this, and then that can pinpoint where the defect is in the metabolic pathway, and hopefully that can lead to a diagnosis. Okay, this all sounds good. However, really, at the moment, metabolomics and lipidomics is really yet to be exploited by the NHS. It's not really moved into the NHS yet. We do have really good expertise in UK universities in metabolomics and lipidomics, and really what we need to do now is to translate the expertise that we've got in the universities into the NHS. So really go from bench to bedside. So at the moment in Swansea, we're doing a little bit of this. We're working with a clinic in Cardiff, which is called a Syndrome Without a Name clinic. So this is people who are in the middle of the diagnostic odyssey at the moment. They've got a disease, but the clinicians can't identify what that disease is. So we're looking at patient samples and trying to help the clinicians to make a diagnosis. We're also working with other hospitals looking at these variants of unknown significance, when there's a mutation but we don't know whether it's going to be pathological or not. So we're helping to determine whether these variants of unknown significance are significant or not. Another thing which we can do is monitor response to therapy. And finally, panels of metabolites can be used in newborn screening. So this is something that's really being pushed in the USA and Netherlands, and the Netherlands really to improve the diagnostic of rare diseases by newborn screening. Thank you.
Thank you so much, Bill.
Good morning, everyone. Thanks a lot for the organisers for inviting me and for this great conference. My first time here, but it's a very inspiring conference. I'm really looking forward to talking to everyone later. So I'm by training a computer scientist, and I work at the UCL Institute of Ophthalmology and Moorfields Eye Hospital. I work on a rare type of disease called inherited retinal diseases. These are just some examples, as you can see in the background here, of what these retinal diseases look like when you image them through the pupil and you look at the retina at the back of the eye. So we've heard some great talks about transcriptomics, proteomics, metabolomics, proteomics, and really I'm at the end of this. I'm looking now at the phenotypes and what is the impact of these molecules on the phenotype. So what you've got here in front of you is on the left-hand side, you've got some examples of retinal scans of patients who have a rare monogenic condition, and the labels underneath are the gene names which are affected in these patients. So ABCA4, for example, is a gene name, EFEMP1, and so on. And what you've got on the right-hand side is a schematic of the cells in the retina. You've heard of cones and rod cells, probably. Everyone has heard of those. So these are types of photoreceptors, which are light-sensitive cells. And
The labels, for example, IBCA4, RHO, and so on, that you can see there, these are the genes that are predominantly expressed in these cell types. So depending on the genes and the cell types in which they are expressed, the resulting phenotype in the retina can be very different. And this is something I'm trying to identify these patterns using AI, so that given a retinal phenotype, using AI, we can try and identify just from the image what gene is likely to be causing the disease. Now, there's over 270 genes which have been identified so far. So it's a very heterogeneous, the inherited retinal diseases are very heterogeneous conditions. So I'll be referring to genetic diagnosis as identifying the disease-causal gene. So I just wanted to give you a bit of background on this specific group of conditions called inherited retinal diseases. We estimate there's around 30,000 individuals who are affected in the UK. The number is likely to be even more. These are lifelong conditions, and they have a significant socioeconomic cost to the UK. And as Gemma mentioned earlier, we have a lot of undiagnosed patients. So even in leading centers that specialize in these conditions, we have at least 40% of patients without a genetic diagnosis. And it can take a very long time to get a genetic diagnosis, especially if patients live in remote parts of the country which do not have access to these centers of expertise. The benefits, everyone here obviously knows about the benefits, but I thought I'd just go briefly over them. So obviously getting a genetic diagnosis can help in terms of psychological support, prognosis, also for family planning reasons. Also, there's some serious conditions, for example, Batten's and so on, more sort of life-threatening conditions that affect more than just the eye, syndromic conditions, that can be detected from retinal scans. So being able to identify those genetic diagnose those is quite important is very important for patients. And also now we've got some treatments, some gene-targeted treatments. Some of you might have heard of Luxturna and RP65 as a gene therapy. There's likely to be one more hopefully coming soon for RPGR, for another gene. And also for the NHS, obviously, it's very beneficial to know, what to diagnose correctly the patient and manage the patient properly. So how can AI aid the diagnosis of inherited retinal diseases? Well, there's two main ways. Firstly, it can help in guiding and assisting the clinicians in their diagnosis and when they're presented with a patient. So what you've got here on the left are 3 types of retinal scans using different imaging techniques. The first 2 are views from the front of the eye, and the one at the bottom there is a cross-section. So being able to look at these images and try and infer what is the likely gene can help give a suggestion to a clinician on what kind of gene diagnosis, what kind of genetic test, could be done. So in the UK, we've got whole genome as a default, more or less, now, so maybe that's not going to make a big difference in the UK, but in other parts of the world, there might be different types of genetic tests, or you might not have, in more resource-poor environments, you might not have enough to genetically test everyone, so being able to know which patients to genetic test might be helpful. And also, we've heard that we have a lot of these variants of unknown significance. A lot of these patients we are unable to really identify the genetic diagnosis for, and that could be for a range of reasons. They were mentioned technical reasons, but also reasons of not having enough knowledge or enough, for example, knowledge here of the phenotype of the patient. So being able to get a gene prediction from a retinal scan can help in these variant interpretation pipelines, which, as you can see here on the right, historically have been using clinical phenotypes, so clinical labels, rather than images. So just describing the patient and getting a clinical label from the doctor rather than actually giving the image as an input. So that's two of the main ways this can help, and how I'm just going to talk briefly about how we built this AI system. So what you've got here are the 3 different bar plots represent the 3 data sets of imaging that we got, different types of imaging that we got from our cohort of patients at Moorfields Eye Hospitals. There's around 3,000 of these patients in total, 3,000 families, but we got data from around 2,000 of these patients, and this covers around 36 genes. There's much more genes, but we just don't have sufficient amount of data for some of these ultra-rare conditions. And as you can see from these bar plots, this is each bar represents the number of images we've got for a specific gene condition. So this is the training data set we used. The images are labeled with the gene. So we use this to train the AI system, and we focus on the 3 different types of imaging that I mentioned. The 2 from the front, which are the autofluorescence imaging, which uses autofluorescence image and can show you a different parts of the retina. The infrared also, that gives you a different sort of view of the retina and the cross-section. And combining these predictions gives you a combined result. So the advantage of doing this is, for example, some hospitals might only have 1 type of imaging, or you might not have all the images from all the patients. So being able to look at them separately has advantages. So the results we get are a top 1 accuracy. So out of 36 genes, 66% of the time we get the correct gene as the top 1, and 85% we get it within the top 5. So this is we did this in our internal data set at Moorfields, but when you develop these AI systems, you really need to validate them externally in other data sets, and that's what we did. We went to other sites, and we asked to get images from patients with inherited retinal diseases, and these are the results we obtained. So across the board, they are quite consistent results. And if you think that 66% isn't a very high number, I agree, 100% would be better, but it is a very difficult task. And just to illustrate that, we also gave it to some expert ophthalmologists. We gave them images, and we asked them to see whether they could guess the gene. And top 5 accuracy was 32% versus the IT genes on that specific data set being 70%. I mean, it's a very difficult task to do, and it's not really something that people usually do in isolation. So it's nice to have a tool that is able to do that. And another nice thing about using this tool is that what you can do is you can start looking at the similarity between the different conditions and building groups based on this. So what this is a clustering diagram here. And basically, genes which are close to each other means that the retinal phenotype is similar. So from this, we can already see some groupings which make sense, but also we can also discover potentially other groupings. And this is quite important because, as you probably know, doctors tend to label things and put things into boxes based on historical textbooks and so on. And being able to do this in a more sort of data-driven way could be quite beneficial. So in conclusion, the IT-Gene AI predicts the 36 most common inherited retinal diseases to a top 5 accuracy of over 80%. It can help with phenotypic similarity between patients. And obviously, but there's a lot more work to do in trying to increase the number of genes that we can identify, and also doing more clinical evaluation. But we hope that eventually we'll be able to distill this expertise in going from phenotypes to genes across more places in the UK and in the world. So thank you very much.
So thank you very much to all our speakers. We have not been helped by the weather this morning, and we got started a little late, so I'm going to just be very, very brutal, and instead of having a panel discussion, we're going to take a question from the audience, but you can catch all of our speakers, obviously, during the breaks throughout the day. So does anybody have a question?
Hello, my name is Matt. I don't need a microphone, it's loud. I've got a weird sleep disorder, rest sleep disorder, so it manifests in the brain. How can your sort of technology material?
That's a great question. What about the brain? How do you get to the brain? I'm going to say, Jamie, can you get to the brain?
So one of the things that we've tried to focus on is understanding kind of which tissues are appropriate to replicate what's elsewhere in the body. And so, like Nico, kind of my main research interest is in the retina, and the retina is an extension of the brain, yet we could probably see 30-40% of those genes expressed in other tissues. And so when we look at blood, actually bloods can be quite a bad thing because it's just dominated by things like hemoglobin that get transported everywhere. But there's other approaches, so we can transform some of the cells in blood into different things in the lab. We can also take things like skin scrapings and look at fibroblasts, and you can go to the extremes of making things like induced pluripotent stem cells, so applying particular chemicals to cells that have been extracted, getting them to this really kind of immature state, and then there are technologies to kind of differentiate those into different cells. So whilst that might not replicate what's there in real life going on in your brain, it goes some way to kind of understanding the development of those processes in some instances, but also the fact that we've got some of these technologies, we can actually detect whether we find particular genes being expressed or represented in those particular cell types, and so we can say how appropriate a particular method may be to look at particular things. So it's a little bit of a woolly answer. It's a very difficult situation. These things aren't stable. They're changing all the time, but we can get part of the way there by applying different types of techniques.
So we might be able to make brain cells out of other cells and do it that way. We might be able to use CSF and do it that way. And I guess you just have to see what you can find in the blood in terms of...
Yeah, absolutely. It is definitely a challenging situation, but as Jamie said, there's sometimes mechanisms going on in other tissues that we're not exactly, that may mirror some of the kind of defects going on in specifically the brain tissues, and if they end up in blood, sometimes you can pick them up, but it's certainly a challenging situation.
This is Rare Summit, and this is where these conversations happen, so please do pick up with each other and think about how different technologies can be applied to your problems. Thank you. All right.